Hello, I'm Gustavo de Souza. I'm a professor here at the Brain Institute and the Bioinformatics Multidisciplinary Environment at the Federal University in Natal, Brazil. And I'm going to talk to you briefly about a tool that we developed here called Proteogenomics Viewer. Proteogenomics is an approach which you can use peptide sequence information, mostly collected using proteomics approaches, to align it to genomic information in order to improve genetic models. For example, you can collect, you can use the peptide sequence to validate if a coding region of a genomic region on the is coding or not, or you can use, for example, peptide information to characterize splicing events and different protein isoforms. Our aim here was to develop a very user-friendly tool where you could upload your proteomics data and you could easily retrieve all the information needed for proteogenomics, and this would be, again, easily accessible through a couple of small, few clicks, and you could see all the information you need to get like a proper validation of your data. The tool itself is divided in two parts. Of course, you have the script itself in Perl, and that is going to be mostly used by the people that want to upload their own data. And it can accept, the kind of input it accepts is TXT files, which you can get from your peptide search engine, or you can also collect, use m.mzid files from the PSI standard from the Human Proteome Organization. The other part, of course, is what you see here, is a web browser interface, where you can be like a proteogenomics researcher and use this again to validate your data and even to report your data. Or on a more broad audience, you could be like a biochemist or a geneticist and you're interested in a peptide of relevance. You can access our viewer on the web address that is going to be available in the publication, and you can see the identifications that we did on our own analysis for the publication. For example, I here show only a small example of a protein called LAMC1, laminin gamma chain, and you see that we get public proteomic data, used a database which we predicted uncommon isoforms or novel isoforms based on half-sec data, together with reference sequences from UniProt. And you can see here the data is loaded. You have the reference protein on the bottom here. You can mouse over, it's going to show you the name, but of course for the video it's not going to show properly. We can select the peptide, which is going to be highlighted in the primary structure and the sequence of the protein. In this case, this peptide here shows an exon skipping of this exon here, because you have the alignment with the genomic information. And with a couple of clicks, you can see the quality of the identification. You can see that it has if you don't understand the spectra annotation, you can still see that it has a very good scoring and a very low error probability calculation. You can see that the quality of the identification is good. And also, importantly, you can see in which data sets that protein is present, that peptide is present, and at which levels it was detected by the instrumentation. And you can also see that that peptide in question does not align to other sequences on the genome. So you see that it only aligns to the gene that you are analyzing. So of course this gives you extra confidence of the identification you performed. So our most important for us here in developing this tool was that it not only gives you like a genomic browser interface for peptide identification sequence, but also all the indexation of the peptide sequences within the genome are provided by the tool later on as an output from the tool. So you can collect this indexation and you can do your own post-analysis. For example, in this case, we use it only illustrative. You use it to detect uncommon novel isoforms which has loss of transmembrane domains. So it's like we use this indexation to quickly have an access to this kind of information. So that was pretty much what I wanted to tell you. Thank you for your attention. And if you want to know more details about this tool and you want also to contact our group, please check our publication at Bioessays.